Welcome, everyone. Today, we're diving into the world of RNA extraction. We'll discuss the working principle, steps involved, and its significance in clinical diagnostics. We'll also delve into various platforms used for RNA extraction. Let's start now. RNA, or ribonucleic acid, is a type of nucleic acid present in all living cells. RNA plays a critical role in the central dogma of molecular biology, which describes the flow of genetic information within a cell. The history of RNA extraction techniques can be traced back to the mid-20th century. The process involves isolating RNA from biological samples such as cells, tissues, or viruses. The primary goal is to obtain a pure and concentrated RNA sample free from contaminants and other cellular components. This purified RNA can then be used in various downstream applications, including gene expression analysis, transcriptomics, and the detection of pathogens. Now, let's take a closer look at the working principle of RNA extraction and see how this vital process helps us unlock the mysteries of gene expression and function. The working principle of RNA extraction involves breaking open the cells or tissue and separating RNA from other cellular components such as proteins, lipids, and DNA. There are several methods for RNA extraction, but most of them follow a similar workflow. The first step is sample collection. Collect samples according to the specific requirements of the tissue or cell type being analyzed. This may involve using sterile techniques, RNase-free equipment, or specific collection devices, for example, swabs for respiratory samples. It is important to collect samples promptly to minimize RNA degradation. The second step is cell lysis. A lysis buffer, which typically contains a detergent and a chaotropic agent, is added to the sample. The detergent disrupts the cell membrane, and the chaotropic agent denatures proteins, including RNases, enzymes that can degrade RNA. This step helps to protect RNA from degradation and inactivate cellular components that could interfere with the extraction process. Then, the lyse sample is homogenized to create a uniform suspension. Homogenization can be done using a variety of methods, including pipetting, vortexing, or using a mechanical homogenizer. The next essential step is RNA separation. The method used to separate RNA from other cellular components depends on the extraction technique employed. One of the most common methods is phenylchloroform extraction. This technique relies on the differential solubility of RNA, DNA, and proteins in water and organic solvents. The sample is mixed with phenol and chloroform, and after centrifugation, RNA petitions into the aqueous phase, while proteins and DNA remain in the organic phase or at the interphase. The aqueous phase is then collected for further purification. The next step is RNA precipitation and washing. The RNA is often precipitated using ethanol or isopropanol and a salt, for example, sodium acetate. The precipitated RNA can then be pelleted by centrifugation. In the RNA extraction process, RNA is precipitated into a solid, granular substance through methods such as centrifugation, which is referred to as an RNA pellet. The RNA pellet is washed to remove any contaminants, usually with a 70 to 80% ethanol solution. This step helps to remove any residual salts or impurities that could interfere with downstream applications. Next is RNA resuspension. After air drying the RNA pellet to remove any remaining ethanol, the RNA is resuspended in an appropriate buffer or nucleus-free water. It is important to use RNase-free reagents and consumables to prevent RNA degradation. Once the RNA is resuspended, then comes the step called RNA quantification and quality assessment. RNA is assessed using spectrophotometry, for example, measuring absorbance at 260 nanometers, fluorometry, or a bioanalyzer. It is essential to check the RNA's integrity by examining the RNA integrity number, or the RIN, or by running a denaturing agarose gel electrophoresis. At last, storage. Store the purified RNA at -80 degrees Celsius for long-term storage or at -20 degrees Celsius for short-term storage to maintain RNA integrity and prevent degradation. The following is a schematic diagram of the complete RNA extraction process utilizing phenol chloroform extraction. Over the years, various platforms have been developed to streamline and automate the RNA extraction process. These platforms range from traditional manual methods to cutting-edge automated systems. Let's explore some of these options. Manual RNA extraction. Traditional methods involve using basic laboratory equipment and reagents to perform the RNA extraction process. Phenylchloroform extraction method has been discussed in detail above. This method, though time-consuming and labor-intensive, can yield high-quality RNA. Now, let's get to know some of the other common RNA extraction kits. Column-based kits. These kits usually use silica-based columns or spin columns to selectively bind RNA. The sample is passed through the column, allowing RNA to bind to the silica while other cellular components are washed away. A series of wash steps are performed to remove contaminants, and then the purified RNA is eluted with a low ionic strength buffer or water. These kits offer a faster, more consistent, and reproducible method of RNA extraction. The magnetic bead-based kits. Magnetic bead-based kits are widely used for RNA extraction and purification due to their ease of use, high efficiency, and scalability. These kits utilize magnetic beads coated with a specific ligand that binds RNA. The beads are mixed with the lysate, and the RNA binds to the beads. A magnetic field is then applied to separate the beads with the bound RNA from the rest of the sample. After washing the beads to remove contaminants, the purified RNA is eluted. Automated systems. Several automated platforms have been developed to further simplify and speed up the RNA extraction process. These systems often use magnetic beads or other solid phase extraction methods coupled with robotic liquid handling and automation to minimize manual intervention, reduce errors, and increase throughput. Examples of automated RNA extraction systems include the Maxwell RSC instrument and the Thermo Fisher Scientific Kingfisher Flex. RNA extraction plays a crucial role in clinical diagnosis and research. For instance, it enables the identification of pathogens in infectious diseases, such as viral infections, for example COVID, through the detection of their specific RNA sequences. Additionally, RNA extraction is fundamental in the study of gene expression patterns, which can provide insights into the underlying mechanisms of various diseases, such as cancer or neurodegenerative diseases. Furthermore, the analysis of circulating RNA, such as microRNAs, has shown great potential as a non-invasive diagnostic tool for monitoring disease progression and response to therapy. Applications. Both DNA and RNA extraction have a wide range of applications in various fields, such as scientific research, clinical diagnostics, and forensics. However, their specific applications differ due to the distinct roles of DNA and RNA play in cellular processes. DNA extraction is primarily used for applications involving genetic information analysis, such as genetic testing and screening, infectious disease diagnosis, oncology and personalized medicine, and forensics. RNA extraction, conversely, is mainly used for applications focusing on gene expression and cellular processes, such as transcriptomics, functional genomics, RNA-seq, real-time quantitative PCR, or qPCR. As you can see, while both DNA and RNA extraction share some commonalities in their working principles, their specific applications differ due to their unique roles within cells. The interplay between DNA and RNA extraction techniques has transformed research in diverse disciplines from medicine and agriculture to conservation biology and forensic science. In conclusion, understanding the DNA and RNA extraction is essential for appreciating the intricate tapestry of life. So as you venture further into the captivating world of nucleic acids, remember that DNA and RNA extraction are the gateways to understanding our genetic heritage and unraveling the complexities of life. Thank you for watching.